Enzyme inhibition. Some parts of an enzyme are the active site and the allosteric site. An active site is the key location on an enzyme that a specific substrate will bind to. An allosteric site is a site that is separate from the active site that another substrate can bond to in non-competitive inhibition. Allosteric inhibitors modify the active site of the enzyme so that the substrate binding can no longer happen. In the normal process, the substrate ATP and phosphate bind to the active site of the phosphofructokinase enzyme and are turned into ATP by the enzyme. As we can see in the second reaction, when the concentration of the ATP is high enough, an ATP molecule will bind to the allosteric site of the enzyme. This changes the shape of the active site and prevents more ATP from being formed. Competitive inhibition is a process in which a substrate molecule is prevented from binding to the active site of an enzyme by a molecule that is very similar in structure to the substrate. In the first reaction, we can see a substrate binding to the active site of the enzyme. The enzyme then rearranges substrates and breaks it down into multiple new parts. In the second reaction, we can see that a different substrate binds to the active site, which prevents the original substrate from binding and being broken down into new products. Both competitive inhibition and non-competitive inhibition are examples of feedback loops. There are many different categories of enzymes. Here are just a few. A hydrolase enzyme breaks down substrates. It specifically does this through hydrolysis. It adds water to split chemical bonds in a reaction. This type of enzyme is usually used to break down larger molecules into smaller ones. A lyase enzyme is a specific type of enzyme that breaks down a substrate. It does not use oxidation or hydrolysis when it does this, and often the product it makes contains a new double bond or ring structure. The end.